Thanks everybody for joining the call. I was just telling Michela that I've been looking forward to this one for quite some time, basically since the paper came out, because, well, we had not really thought of GBA, GCase, as a mitochondrial protein. So really excited to talk about this. The conversation obviously today was sparked by this manuscript, this beautifully done manuscript by Dr. Michela Deleidi, who is joining us here on the call, which is titled Glucocerebrosidase, I'll refer to it as GCase from here on, is imported into mitochondria and preserves complex 1 integrity and energy metabolism. She has been kind enough to join us from Paris today, and I'll just give a very brief introduction. Dr. Deleidi is group leader at Imagine Institute of Genetic Diseases and also assistant professor at University of Tübingen. Thank you so much for joining us.
First of all, thanks for the invitation, and thanks for the kind introduction of our research and our group. Hi, Andres. So, as you may know, one of our major research interests has been in the last few years since I started my group in Tübingen, has been really to look at mechanisms involved in GBA1 associated degeneration. GBA1, as we know, is the most frequent risk factor, a genetic risk factor for Parkinson's disease, and is probably involved also in some pathogenetic pathways in sporadic Parkinson's disease, and I would also say in the general aging process, along with the key role of lysosomes in the aging. And when we started our research, we really focused, as everybody in the field, on the lysosomal function on GBA, how to link this general lysosomal dysfunction to mechanisms that could be specific to Parkinson's disease, as well as on the other side, to more acute neurodegenerative pathway, which is observed in patients with Gaucher disease. However, during the last few years, we also discovered that despite being a primary lysosomal protein and enzyme, we first described, we and others, because this was already published by the group of Duchenne using GBA knockout mice, that actually the defects, loss of function of GBA is profoundly linked to mitochondrial dysfunction at several levels, including energy metabolism, mitophagy, and ROS production, and several other mitochondria-related pathways. And using induced pluripotent stem cells generated from our patient cohorts in Tübingen at that time, we also proved that mutations in GBA1 are linked to mitochondrial dysfunction. And it was interesting to note that those data show that both gain and loss of function are actually involved in these mechanisms involving mitochondria in these primary lysosomal conditions. And after that, I come now to the recent paper. So we started, this data actually, these findings came out a bit by serendipity, because the major goal of that project at that time was to identify potentially novel targets that could be exploited as chaperones to promote the folding of GBA and the transport of GBA from the endoplasmic reticulum to the lysosomes. And in particular, we were interested in looking at whether different mutations, in this case, the frequent L4-4P mutation, which is very frequent in these patients as well, and the E326K mutation, which is a point mutation found uniquely in Parkinson's disease patients and not in Gaucher, could differentially affect the interactome of GBA. And what we did is to generate inducible Excel lines expressing specific mutations, and looked at how the glucocerebrosidase interactome was affected. And at some point, so looking at the data, and this was done with the TMT mass spec, we were really surprised, because we saw that up to 18% of the interactors of GBA were actually found in mitochondria. So this was an overexpressing model system, and we had to exclude that these data could be attributed to a sort of overexpression-related artifact. So what we did first was also to look in the literature, and we already knew that at that time there was only one GBA interactome study performed in HeLa cells at the endogenous level by the group of Jeffrey Kelly. And what we did is to look and compare our results in our overexpressing modeling system and their results and the endogenous level. And what we were able to see is that, and it was reassuring, is that almost 90% of these mitochondrial interactors overlapped between the two studies. And so this convinced us that actually this mitochondrial role of GBA could have something, could go beyond this indirect effect of GBA on mitochondria. So we looked at different interactors, and we noticed that glucocerebrosidase interacts with mitochondrial proteins involved both in import mechanisms as well as in the mitochondrial protein quality control, including proteostasis and energy metabolism candidates. So what we did, of course, was to, sorry, that's something, okay. So what we did is to conform all these candidates at first in our overexpressing modeling system, but more importantly at the endogenous level. And what we used was induced pluripotent stem cells generated from patients carrying the mutations of interest. So L44P and E32K mutations, as well as their corresponding isogenic controls. And what we were able to see combining these stem cell modeling approaches as well as in silico modeling is that GBA is indeed imported into mitochondria. It doesn't contain a conventional mitochondrial targeting sequence, and this import is mediated by the recognition of the so-called mitochondrial targeting-like sequences. And this import is mediated by the TOM and TIM23 machinery. And once in the mitochondrial matrix, we confirm the presence of glucocerebrosidase within the mitochondrial matrix using different techniques, including mitochondrial isolation, followed by biochemical confirmations, as well as expansion microscopy in neurons. And what we what we show and what we found is that glucocerebrosidase in physiological conditions within the mitochondrial matrix interacts with the complex 1 subunits. It promotes the assembly of a complex 1. However, mutants, such as the L44P and E32K mutations, alter this interaction of glucocerebrosidase with complex 1. And we found that mitochondrial GCase, mutant GCase, is degraded by the mitochondrial quality control mechanisms. So we were able to show for the first time that a portion of GBA in summary is imported into mitochondria and plays a key role in maintaining mitochondrial function. And this is very important, especially if you think and the known role of complex 1 in the context of Parkinson's disease and dopaminergic neuron vulnerability.
Thank you so much, Dr. Deleidi. And as you said, I think when I saw the title and thank you for putting the punchline in the title, because I saw the title and I thought, what? GCase actually gets into the mitochondria, because that's the first time that, at least that I've seen, as you mentioned, quite a few, several other groups had shown impact on mitochondria, but I think all of us, at least myself, if I can speak for myself, had assumed that that happens indirectly through the actions in the lysosome or potentially other, the ER or Golgi or the other two places where we kind of thought about GBA as even being present. Let me take a moment before, and I have a million questions. I hope that what we can do is discuss the data before we get into what this means for Parkinson's disease and translation to potential therapeutics. So let's stick to the data for a moment, but we do have several attendees who are, what I consider GBA experts. So if you would like to chime in at any point, please raise your digital hands and I will be happy to promote you to be a panelist and actually take part in the conversation. I think this is one of the beautiful things about this type of a setting is that we can, the conversations don't have to be limited to the back rooms of conferences where we, talk individually. We can all kind of talk about it at the same time. And with that also, let me introduce Dr. Klein, who wrote this very nice commentary on the manuscript that Dr. Deleidi just mentioned. Dr. Klein, thanks for joining us.
Happy to be here with you and also very excited about meeting Michela. I mean, I'm a big fan of your work and it's always...
Thank you, me too. Nice meeting you. First time actually we meet in virtual.
Exactly.
So Andres, what do you think? I mean, we've thought about GBA as a lysosomal protein. We call them lysosomal storage diseases. We've always kind of, I think, talked about GBA in the context of lysosomes. So I guess what was your first reaction and your comments on the paper, if you can talk about that a little bit?
I mean, when I saw the paper, I said, wow, this for me was a wow moment because to me, it really merges both hypotheses about Parkinson's pathogenesis. And that's why I feel it was so important when I saw it. I mean, there was plenty of evidence first. I mean, I would say that the first big discovery was done, by Ellen Sidriansky, in that New England Journal of Medicine paper in 2009, showing that mutations in one GBA1 allele increase the risk of developing Parkinson's. And then we said, but Parkinson's is a mitochondrial disease, and GCA it's a lysosomal gene. So what's going on? And then, well, all the beautiful work that came from the Crank's lab, Joe Mazzulli, showing this bidirectional pathogenic loop, and then in parallel, all these very, very large amount of consistent data showing that in Gaucher there was a lot of mitochondrial problems, and nobody really put it or linked them together. And we were all thinking that, well, the lysosomal gene is dysfunctional, autophagy is not working, therefore mitochondrial turnover it's slower, and we're getting reactive oxygen species, and so on. So the fact that the beauty of this paper for me is first that it comes from a big question. No, it's a big question, and when you ask the right question, you can get the right answer. Then the second point was that you perform an unbiased study, okay, and from there you identify that that the GCAs at least can interact with many mitochondrial proteins. But the fact that it can interact in a test tube in a cell lysate doesn't mean that it goes to that organelle. So you went further, and you then ask the right question: well, is it really there? Challenging all the conservative ways of seeing GCA's biology. And you were brave, and you did it, and that's something that I very much like from the paper. Okay, because one thing is that they can interact. Then if it goes there, and if it goes there, then well, what does it do in this place? And I really think that you went deep into the mechanistic biology, and of course that it has many implications, not only for Parkinson's, but also for Gaucher. And I think that it's opening questions. From time to time, we're just digging in and we say, okay, probably we know everything about GCAs. We're treating patients with enzyme replacement therapies, at least the non-neuropathic. We know how it's transported through SCARB2. We knew everything, but no, we didn't. And you must be brave to have an open mind and to challenge the classical ideas. So I love what you did, Michela.
Thank you.
Likewise, I think the two things that stuck out for me, thanks Andres for pointing those out, because one is the unbiased approach to it and not just kind of looking under the lamp of what we already know to be important. So that was pretty key. And I think the second thing that stuck out to me was the use of the different models that you used. And I wonder if you can talk about that a little bit more, because the initial model was an overexpression model where, you know, I think you always question how physiologically relevant that is, but then you moved into iPSCs, differentiated into dopamine neurons, which is, you know, obviously very with isogenic controls, which is a really nice way to look at it, and then also organoids. So really kind of hammering home that translational and physiological relevance of this work. Can you talk about that a little bit?
Yes, sure. So, as I said, the use of this overexpressing model, the inducible ECK lines, was a kind of something that we had to do because as I mentioned before, our original question was different, right? We wanted to identify and to study how these mutants, which are linked to different conditions, can differentiate affect the GBA interactome. And as we know, especially mutations such as the L44P mutations severely affect the folding of GCase and the level of the protein, because it's prematurely degraded by the ERAD system. And so it was not possible to go right away and to study at the endogenous level how these mutations, pathogenic mutations, affect GBA1 interactions at the cellular level. And so that's the reason why we had to use this overexpression model system that allowed us to have a sufficient yield of protein to go ahead with our proteomic studies. And I still think that in science, also based on your question, you have to choose the right model. And probably starting with the simplest model helps us identify also some solid targets and to conceive hypotheses. And so, as you said, definitely this model has some limitations. So after confirming these targets in the overexpression system, we had to go into details and to choose a model that was similar, more relevant for the disease. And that's the reason why, also based on our expertise, we turned to induce pluripotent stem cells. And we generated additional lines in order to avoid issues with the non-variability, which is known in the iPSL field, and using isogenic controls. And we did this both in 2D and in 3D model systems, so using midbrain organoids. And 2D again is relevant since we can focus on a cell type specific vulnerability, in this case dopaminergic neurons, even though I think that limiting our analysis to dopaminergic neurons can also be something that we are also moving to other cell types, looking at whether these mechanisms can impact, for instance, immune cell function, which is another important pathogenetic pathways in Parkinson's disease, especially considering the role of mitochondria in modulating immune cell function. So, but what we did in this paper is to use 2D model systems, so dopaminergic neurons, to confirm the import of the protein into and the mitochondrial localization. And so we were able to validate and to describe these mechanistic pathways that link GBA1 to a mitochondrial function, and especially to complex 1 activity. And finally, since, as we all know, we do not really see key pathological features resembling Parkinson's disease in 2D model systems, we employed midbrain organoids generated from mutant lines and controls. And there, what we observed, and it was very important, is that the GBA in long-term culture organoids, we kept these organoids up to 150 days. These organoids carrying GBA1 mutations develop pathological features of resembling Parkinson's disease, including Lewy body-like structures. And we have done additional work after the publication of the paper confirming that these pathological aggregates of alpha-synuclein within midbrain organoids actually resembles in composition, lipid compositions, and other characteristics Lewy body present within the brains of the patients. And also, we were, again, I think it's important to see that what we found is that using single cell analysis and looking at the cell type specific impact of GBA1 mutations, it was very interesting to see that the GBA1 mutations are linked to complex 1 dysfunction at the pathway level and the mechanistic level in midbrain dopaminergic neurons within our organoids. Of course, still, what we have to do, and probably is the most challenging part in all the modeling field, is to show causality. So to prove causality, so to really prove that what we see, the mechanism is there, the localization is there, and the function is there, but we have not proven that this is directly linked to the pathological features that we could observe in our 3D model systems.
Wonderful. Thank you, Dr. Deleidi. I want to get Dr. Klein's perspective on that as well, but it does look like we have a question. We have Kevin Hunt here from Vanqua Bio working on GBA. Did you have a specific question?
I sure do, yeah. Well, thanks for arranging this, and I appreciate everybody's time and talking about this fantastic research. I'm just curious, and maybe I'm oversimplifying this in my head. Do you think that the mutant GCase transport to the mitochondria is a potential compensation to relieve the mutant GCase burden in the Golgi? Is this a way for the Golgi to relieve some of this burden? And was there a relationship between Golgi burden versus the uptake you saw in the mitochondria with the mutants?
Yes, this is a very interesting question. And then it's also, we quantified with all the limitations of the quantification in this model system. And the proportion of GCase, mitochondrial GCase in the mutants and in the wild type cells. And what we observed is that actually we can see more mutant GCase within mitochondria compared to the wild type GCase. So probably, yes, this import within mitochondria can be also a sort of additional quality control mechanism that helps the cytosolic quality control mechanism that is kind of overwhelmed by the mutations. And this is as all this basic mechanism has been described in a few years ago and has been defined as the magic function of mitochondria, where they can support the quality control mechanism in presence of cytosolic misfolded proteins. It would be interesting to see which pathways regulate this mitochondrial import, because so far we have not seen differences in interaction with the chaperones that could be involved in these pathways.
Okay, that's a follow-up question there. You know, with these nice models, have you looked at the effect of an inhibitory chaperone such as Ambraxol? Does that affect does it decrease mitochondrial uptake as you
Yeah,
correct folding?
we are doing that. Yes, exactly. So
Oh, fantastic. Okay. Any previews?
Not yet. Not yet.
Okay. Well, thank you so much for answering my questions, and thank you for the research.
Thank you.
More questions have come in through the chat as well, which I'm happy to ask. But I did want to maybe very quickly, and maybe both you and Dr. Klein can comment on this, is talk about the choice of the GBA mutations, because there are several of the GBA mutations, and then the relationship between Parkinson's and Gaucher's, particularly as it relates to heterozygous versus homozygous mutation carriers. So I think they do tend to behave, well, depending on the model, they can be a little bit different. And there was one question that came in that's related to that maybe I'll just throw into this as well, which is, do Parkinson's cells with GBA mutations accumulate glucosylceramide similar to Gaucher cells, which I think kind of gets to that question as well.
Yes. So what we described, so this is still a bit controversial. So in what we initially described in our IPSL work in 2014 is that what we observed, and actually it was conducted with Pablo Sardi at that time, we saw an increase of glucosylceramide in total glucosylceramide in iPS-derived neurons when comparing GBA1 mutants with severe mutations to the corresponding isogenic controls. We we can't say that there is an accumulation at the substrate. I think it's important to say that. Also, it's like glucose, right? In diabetes, there are some parameters and some thresholds in which you can define the diabetes or intolerance. And what we observe is an increase, but definitely there is not such as an accumulation as it's observed in Gaucher disease cells. And what we have done afterwards, and other groups have shown using also a more detailed mass spec analysis, that probably only some species of glucosylceramide are increased in dopaminergic neurons. Whereas if you look at homozygous biallelic mutants, Gaucher cells or GBA knockout cells, you see really a massive increase of both glucosylceramide and glucosylsphingosine. Having said that, we can't exclude that in susceptible cells, such as neurons, dopaminergic neurons, with aging, even a minor imbalance or change in lipids can actually predispose to disease. So this is something that we can't exclude. And this was also very important, and I conclude also because one of the initial hypotheses we had in mind is that the link between GBA and mitochondrial dysfunction could be indirect, and mostly due, for instance, to this lipid imbalance on mitochondrial membranes. Lipids are very important on mitochondrial membranes as well, especially on mitochondria, but we could not observe an impact of heterozygous mutations as observed in Parkinson on lipid compositions in mitochondria.
Very interesting. Dr. Klein, any comments on that?
Yeah, absolutely. So there is a huge controversy about the lipid buildups in heterozygous, and some groups have shown little increase in glucosylceramide, others in gangliosides, but not in ceramides. And recently, Ricardo Feldman showed glucosylsphingosine and not ceramides. So I believe that this is more complex and probably a subset of heterozygous might accumulate lipids, and others probably they do not. This topic, it reminds me of a beautiful paper also published by Ricardo Feldman in Human Molecular Genetics, I think 2015, where he analyzed iPS dopamine neurons derived from type 1, type 2, and type 3 Gaucher patients. And he saw cellular differences, okay? They all had GBA1 mutations. They were different mutations, and the type 2 particularly, and also the type 3 neurons, they had less number of lysosomes or lysotracker positive organelles compared to type 1. So I think that here there's still some pieces of information missing, and we're still scratching the surface of the problem.
Yeah, and if I may add, I think that also with new technologies such as recent developments in single cell lipidomics, such as the technique also demonstrated by Giovanni D'Angelo at EPFL. We are not probably at the single cell level yet, but it will be very important both in mouse models, but especially in patients' brains, to look really at these changes at the cell type specific level to see whether this is, as you know, as we know, for instance, in immune cells, we have different levels of glycosphingolipids and different metabolism compared to the one observed in neurons.
And
one
also
thing
there
with
is a
that
huge
too,
variability. Sorry, also there is a huge variability among dopaminergic neurons.
Exactly. Yeah. Exactly.
One thing that we did was do computer simulations of the pathway as it relates to lysosomes, not at all in the mitochondrial side of GBA. And it seemed like it was very heavily dependent on expression level of acid ceramidase, as opposed to as to which direction it ends up going, whether you get accumulation of glucosylsphingosine or glucosylceramide or neither, if the levels are kind of, and I don't want to say equal because they are different, but yeah, if you reach that equilibrium. So I think that maybe looking at, as you say, in the single cell, if you can look at single cell, we can actually look at the expression levels of each of these proteins, at least. Activity, that's a whole nother challenge. Let's move on to, well, I guess a couple of things. One, I do want to get into alpha-synuclein and talking about alpha-synuclein, but before we do that, do you think, and there was a little bit of a hint towards this earlier, there have been other manuscripts that have shown deficits in mitochondrial function, and I'm just highlighting a couple of them here. I apologize if I have not highlighted one that somebody here has worked on, but looking at, mitochondrial priming for autophagy, for example, which seem to be affected in this manuscript from the Columbia group. Also looking at calcium dysregulation, notably at the mitochondria, reduced membrane potential, oxygen consumption rate, and then even potentially mitochondrial lysosome context. Do you think any of these can be explained with this new information now that GCase can actually get into the mitochondria? Or do you think, I guess, how do we, particularly for the one where they suggested that there is that mitochondrial, lysosomal function of GCase is not affected, but do you think that this might explain some of these other abnormalities that we're seeing that are not directly complex 1?
Yeah, definitely they are. So first of all, so these two organelles, they are functionally and physically tightly connected, as you've shown from this beautiful paper by Dimitri Cranes, also the original Nature paper, showing the mechanisms linking these two organelles. And I think we still need to work on how these two organelles interact not only at the physical but at the functional level by exchanges, for instance, metabolites or other factors. And this could be definitely influenced by the localization of a GBA within mitochondria.
Dr.
Klein, anything to add to that?
No. I mean, I fully agree with Michela's comment. I also think that another beautiful aspect of your work is that you work with the E326K mutant that doesn't cause Gaucher, but it does cause Parkinson's. I think that was beautiful. I must recognize that I missed the N370S mutation.
Yes.
It would have been beautiful.
I was going to bring that up too.
Yeah, that's time. Yeah, we would have liked to, and it was simply not feasible in terms of, as you know, manpower and also, yeah, the number of lines and controls you have to use. But definitely, and I mean, the scientific reason behind the choice of those two mutations is that we saw them as the opposite spectrum. One causes severe Gaucher disease and earlier a form of Parkinson disease with an earlier onset compared to other mutants, and the E326K, as you said, is not observed, is not found in Gaucher disease patients. It's not definitely a mild mutation because E326K patients have an earlier onset and also more severe dementia. So it would be, for instance, interesting to see also how this mitochondrial aspect could be linked to these clinical phenotypes. And there is also a recent paper from Tony Shapiro showing, for instance, alpha-synuclein related mechanisms seem to be more severe in the E326K mutations. So this is still, I think, very intriguing.
The topic of the mitochondrial lysosomal contact sites also has me thinking about how GCase is actually getting into the mitochondria, and everything that we've had in our models previously has been that GCase is a membrane contained protein and going through the late endosome to the lysosome. And I'm wondering, did you somehow fractionate and confirm GCase in the cytosol, or is it possible that it's getting to the mitochondria through ER mitochondrial contact sites or otherwise?
Yeah, so we can't exclude that this is also a mechanism by which it gets into mitochondria. What for sure we have seen that the import mechanism, and we have also demonstrated this using split GFP models, is the recognition of internal mitochondrial-like sequences, the HS70 pathway, the TOM pathway. So this is one of the conventionally important mechanisms. We can't exclude that other pathways may be involved in this mitochondrial localization, but we have not looked at it. For sure, due to this tight interconnection between these two organelles, these three organelles, this could also lead to this mitochondrial localization.
I do have to second that as well, because this part, if I can point to the picture of, GCase just going into the cytosol, how it would be nice. And I know each paper that comes out with really interesting data leads to, I think, 10 more questions, but how is it actually getting into the cytosol? I think as a follow-on to Andy's questions, that would be an interesting one to mechanistically dissect.
And how it's regulated, because as I said, we see different, for instance, mutants the levels of mutants are higher compared to wild type. But we have not seen any differences, at least at the interaction level, with some of the candidates involved in these pathways. So it's not probably regulated by different interactions.
Thanks, Andy, for the question. We also have Peter Lansbury, who knows a thing or two about GBA.
So first of all, I really like the way you talk about this. You use really precise language, and I think that kind of science is really missing. But I want to take a little bit. So what if we just say that this is part of the problem? I think you make a strong case that it's part of the problem. And I think the L44P is a really interesting one because the rate of progression is so fast compared to N370S, which is actually slow. So I think if you look at the Sanofi clinical data, E326K is right in the middle in terms of rate. And of course, the rankings are different if you look at risk. So maybe there's a combination here of, this is not a evolved process. It could be, it's just a mess that happens when the protein can't fold or function correctly. So what if there's a mixture where you see one feature of the enzyme driving onset and another feature predominantly driving progression?
Yes.
I do think it would be really important, A, to to study N370S in your system because it's so unusual. If you look at that, and you don't have to just look at the Sanofi data, by the way. If you look at the Fox database, that clearly shows that, although it's smaller numbers, that the N370S is a slow progressing form of GBA-PD. So that begs the question of a mixture. So I wonder if there's any data out there where people have looked at either risk and or rate of progression and correlated that to some sort of mitochondrial marker that you think should be affected by your mechanism. I don't know what that would be, whether it's a biomarker or a functional biomarker, or if that study could be done where you could start to separate potentially a GBA-PD into patients who are primarily driven by activity and patients who are primarily driven by folding. Do you have any ideas about that folding and or what the consequences of misfolding?
Yeah, this for sure. To the best of my knowledge, this has not been done, and I have not seen not even preliminary data at conferences or not even in the Michael J. Fox Consortium. But definitely, this was also one of the driving questions of this study, to see whether we could observe, we could see some targets that could be even explored, as you correctly said, as biomarkers to even further stratify this genetic group of Parkinson's disease patients. I think this should be done. To the best of my knowledge, there is no ongoing study. And as I said, definitely the effort for P is faster, but what is I also find extremely interesting is that the E32 patients are more demanded. So this is also something important that we should keep in mind, and probably also to look at the mechanistic level, at how this mutation affects probably even other neuronal cell subtypes, and at the single cell level, and as I said, on the biomarker levels, to see whether we could differentiate lysosomal versus mitochondrial markers that could give us information about this different rate of progression.
Yeah, no, that's a really smart that dementia issue is critical. I don't think Fox Foundation has a lot of good data on onset or rate of progression. I think all they have is MMSE scores, which are next to useless.
Also because from a clinical point of view, it's something even easier to follow, right? To follow up and to look at clinically as rather than motor symptoms. And
oh yeah, no, I think what you should do, I think what should be done, which would be interesting, and we're now getting to, you know, what I think is the issue here is there's no such thing as Parkinson's disease as a single entity. But I think, and we did this with GBA, but we didn't do it with individual mutations, which was a mistake. But we looked at the Fox, and we looked at progression of each component of the UPDRS3, and they do not correlate. And the lack of correlation depends on the mutation. So as you said, you could view cognition as a symptom of Parkinson's disease, although it's not sort of directly in UPDRS3. But even if you just look at tremor, for example, tremor doesn't progress at all in GBA-PD patients, that at least in the Fox collection that we looked at. It just generates noise. Some patients progress more on gait. These are things that neurologists know anecdotally. But I do think that would be interesting too, because I think you could have a cascade of issues here induced by problems with GCase that affect different cells differentially, which affect different symptoms differentially. And I think it makes a really big difference when you're thinking about designing a trial, first based on what the mechanism of your drug is. So the drug that we developed has no effect on folding or stability. So we're going to be missing, we'd like to figure out a way to exclude the patients that have this type of mechanism that you defined going on. And I think when you're looking at rates of progression, you want to find a patient population that progresses very rapidly. I think L44P is, to my mind, it's the best patient group to look
Yeah.
at. If you could do a single genotype cohort, which would be possible in some countries. And I think that could be because they're purely this mechanism, right? So I think this kind of thing is really important to get the word out that it's not good enough to just look at GBA genotype. It's not good enough. And there's no reason it should be good enough. That gets you started. But I think this is really important for that reason.
And can I use this opportunity, Peter, thanks for the comments here, which take us right into the I guess, final portion. And we might have to do another call just to talk about synuclein, because I do think it's an important protein. Obviously, you can't really talk about Parkinson's disease without talking about it. Maybe we have time at the end. But I want to talk about translation, right, for patients. This is a really important question. There are several things in the pipeline at the moment for GBA specifically, which is nice. There's gene therapy. We have two of the companies online here who have worked on activators/chaperone molecules. And there are other things as well. I know, Michela, you've looked at mitochondrial specific things, and some of them are supplements that are available, people can take, like NAD plus supplementation, for example, nicotinamide riboside or nicotinamide mononucleotide, NMN and NR. So I wonder if you can comment, and I know this is a lot, but I wonder if you can comment on what this might mean. And, we would have put Venglustat here as well, but, that one already didn't work. So if mitochondria were the important part, you would expect that it wouldn't work. But of course, it doesn't necessarily mean that that's the important thing also. But can you comment on the various different therapeutics in the pipeline and what you think might be promising?
Yes, so this is a very important part. So as I said, that one part, so from a very basic mechanism level, I think it's important to show causality. Probably it's the most challenging part in this field, but at the same time, how can we target these? So as you mentioned, there are different approaches. What we have done in the past, and it was to use this very generic approach using NAD boosters. And in the view also of these recent findings, I think that there are even novel NAD boosters that are under investigation at the moment. It's important to note that this boosting NAD and increasing NAD levels also has an important impact on mitochondrial quality control, mitochondrial proteostasis, and cytosolic quality control. So this can be an approach that I think should be further evaluated, and that's what also we are doing in collaboration with other groups working on NAD. And I think that I see it as a multi-therapy approach that should really target both the gain and loss of function of this enzyme, definitely exploring the function and the role, the effect of chaperones, including but not only ambroxol, and considering also gene therapy and gene editing approach, probably more in the Gaucher disease field. So I don't have an answer right now. I think, as we just discussed, it will be important to go for a multi-therapy approach, choosing the right patients, stratifying them even better than we did in the past, and to have really more relevant clinical endpoints that could give us, along with biomarkers, targeted biomarkers. I don't have an answer. Definitely we are exploring these three approaches: mitochondrial, cytosolic quality control, and gene therapy / gene editing in our in vitro modeling systems.
Beautiful, beautiful. And I will say we should talk about the mitochondrial biomarkers, because that's definitely an area that's of interest for us. Of course, I didn't ask about mitophagy enhancers as potential, which I'm sure I will hear an earful for from my team on. Perhaps you can comment on that as well. But yeah, as far as mitochondrial biomarkers, I think that is an area that several people are actively working on. So I think we can all kind of come together, and Fox Foundation did put together a group last year. I think we can maybe add some more of us into the group this year who can help with that. Dr. Klein, any comments on the therapeutic side of things?
Yeah, absolutely. I mean, from the data from the interactome, GCase interactome, I think that there are other stuff that you haven't explored yet, and they are very interesting. Well, yeah, it also pointed out to the NADH pathway, which you already covered it, but also to the glutamate receptor pathways. And we all know that glutamate excitotoxicity mediates cell death in many neurological conditions. And that was an area that we explored in the past. I mean, when we treated Gaucher mice, the neuropathic Gaucher mice, with memantine and also with other NMDA receptor antagonists. We just published memantine, but it also worked with 2 or 3 more, which is beautiful from my perspective because it somehow merges the data. But the message is not about our data. It's about that. I think that there is a lot more to explore from your own data. I mean, you were very wise picking mitochondria, but I think there's much more there to explore.
I agree.
And there are questions in the chat. Maybe we can just do a like rapid fire, if I may. We can just give very quick answers because we only have 2 minutes left. How do you address the concern that iPSC neurons and organoids contain young neurons as you reset the age, but PD is an aging disease?
Yes, for sure. And indeed, as I commented, we do not observe pathological features in 2D. In organoids, at the same time, probably due to the conditions that replicate a sort of stress and accelerating aging, we were able to observe pathological features of PD. Still, I would never compare an organoid with an age Parkinson's disease brain. I see it as a bottom-up, as a modular model and a bottom-up seeking mechanism approach that could be useful also to identify targets and to test them.
Fantastic. Can you estimate the fraction of endogenously expressed GCase that's localized in the mitochondria relative to the amount in the lysosome and ER?
Yes, we found approximately 1% of GBA within mitochondria, as I said, with all the limitations of cellular fractionations and quantification on Western blot. Yeah.
Interesting. And then can you comment on the form of GBA1 that's found in mitochondria? It would appear to be the lysosomal endo-H insensitive form.
These we have not done and we have not characterized it. Also, yeah, it would be interesting to see whether it can still have any residual enzymatic activity within mitochondria, which I doubt. But it would be definitely using, for instance, activity-based probes.
Thank you. We did answer the, I'm skipping some of the questions that we sort of answered already. Just one more. Have you tried any methods to detect GCase presence in mitochondria using proximity biotinylation, using BioID or Apex2? Have you used GCase Apex2 in electron microscopy?
No, we are doing some study. I've not data yet. We are currently using Apex2 to further characterize these interactions.
Fantastic. I will end the questions there, but send it back to you both for any final comments.
Thanks for the kind invitation. It's the first time also talking to Michela. I had the chance to talk to Peter like 5 or 6 years ago in Boston. So I'm very happy, and I'm very happy that we're still open-minded to say, you know, there is more here. I believe that with ERT. The research slowed down in the Gaucher field, at least, and now we are re-exploring and keeping an open mind. So I'm very happy for this.
Yeah, thank you for keeping an open mind, and thank you, Dr. Deleidi, for being as was said, being brave enough to go after the mitochondria, even though everything so far has been lysosomal. It will definitely be interesting to see the translational impact of this research, and we look forward to seeing much more from your field.
Thank you so much. It was great. And yeah, looking forward to discussing more about this.
Thank you all. Have a good rest of your day.
Bye. Bye. Bye.